This is part 2 of our story on heredity. In the first discussion, what we mentioned was the fact, the belief that genes were controlling our life, and that was the belief in genetic determinism. However, back in about 1967, I started to see different results in my studies on cells than what I was teaching medical students. While I was teaching students that genes control life, in my lab, a different story was unfolding. That's when I was growing stem cells in tissue culture. And stem cells are like embryonic cells, and I put one cell in a dish all by itself, and it would divide every 10 or 12 hours. So first there was one cell, then it doubles to 2, and then double to 4, 8, 16. It keeps doubling. And at the end of the week, I have 30,000 cells in the Petri dish. What's most important in this research, though, is all 30,000 cells are genetically identical because they came from the same parent. This is called cloning. Now I have 30,000 genetically identical cells. I split them into 3 different Petri dishes. And I grow cells in something called culture medium. Culture medium is the laboratory version of blood. So when I make culture medium, I look at what are the constituents in the blood of the animal I'm using. For example, if I'm growing mouse cells, I look at what are mouse bloods made out of. What is the chemistry of mouse blood? If I'm looking at human cells, I say, what's the chemistry of human blood? And then in the lab, I recreate that chemistry when I make what is called growth medium. So growth medium is the laboratory version of blood. But since I am synthesizing in the lab, I change some of the constituents a little bit. So I have 3 different dishes, all with genetically identical cells in them, but in each dish, I give them a different environment. That's the culture medium, the equivalent of blood. And each different dish had a different combination of chemistry in the culture medium. So in environment A, culture medium A, the cells form muscle. In environment B, the cells form bone. And in environment C, the cells form fat cells. What was the point, and this is very profound, was that all the cells were genetically identical. The fate of the cell wasn't determined by the genes. It was determined by the environment in which the cells were living. Well, this is completely different than the story I was teaching in medical school, that genes were turning on and off, controlling life. Now I find out in my research that the environment is controlling the genetics. Well, trying to find out how that worked led to an understanding. I said, how do I know what is going on? How can the environment control the cell? Well, remember, we go back to our story of proteins. And I say a protein is a molecule that responds to an environmental signal by changing its shape. So a molecule can have shape A, and when a signal in the environment complements that protein shape A, it will cause the shape to change into shape B. And so as the protein moves from shape A to shape B, movement is what is creating life. So life comes from proteins responding to environmental signals and changing their behavior. So signal plus protein equals behavior. Well, the first important point is the study is that the signals come from the environment. But the next most important thing is the signals from the environment have to get inside the cell. And to do that, that means that the signal crosses the cell membrane, the skin of the cell. And at some point, then the skin of the cell must be reading the environmental signals and controlling the behavior. So we start to look at what is the nature of the structure. What is at the interface where a signal from the environment hits the cell? Well, in this model of cells in a petri dish, I put in chemical signals in the form of little physical molecules that you see floating down in the video. And the signals stick to the surface of the cell. And the significance about that is the signal is from the environment. But when you look inside, you see this, that the signal from the environment is translated or transmitted by the membrane from the outside to the inside. And when the signal comes inside the cell, remember, signal plus protein causes movement behavior. So the membrane of the cell, the skin of the cell, is the interface between the environmental signals and the proteins that will respond to those signals. So part 1 of our understanding is signals come to the surface of the cell, and then are transmitted through the surface of the cell, where they engage with proteins and create behavior. This is the basis of a new science called signal transduction. This is the area of science that is actually following how a signal from the environment works its way through the cell and connects all the activities, how signals control behavior. So that's called signal transduction. However, if the protein that I need is not in the cell to make the behavior, and the signal shows up and I don't have the protein, now what? The signal is transmitted from the membrane, goes through the cell and into the nucleus of the cell. And in the nucleus of the cell, the signal selects the gene blueprint needed to make the protein so I could have the behavior. So that part is how a signal goes from the membrane into the nucleus and controls genetics. Well, that's part of signal transduction, but it's specifically called epigenetics. And epigenetics is the science of how environmental signals control genetic activity. So while we used to see in the books, and still do in many books, that the nucleus of the cell is the brain of the cell is not the brain, is not the nucleus. The brain of the cell is the membrane, the skin of the cell. Biologists didn't even know cells had membranes until the late 1940s, when the electron microscope revealed the presence of a structure that had dark, light, dark, a very thin structure that you can't even see in the light microscope. So until there was an electron microscope, biologists didn't even know cells had membranes. But that little dark, light, dark layer going across the surface of the cell in this electron microscope picture is the structure of the membrane. And when scientists looked at the membrane structure, and they said, it's so uncomplicated, it's so simple, it can't be doing very much. So they looked at the behavior of the cell membrane like plastic wrap that would hold the contents of the cell together. So the membrane seemed to have no more function than just a skin to hold the cell together. But what was important is, in their pictures of the microscope, they couldn't see something else, that the dark, light, dark structure, that dark, light, dark banding, is due to the organization of molecules called phospholipids. And it's a double layer where it's like a mirror image, where one layer of phospholipids faces the other layer of phospholipids. And this is the membrane of the cell. And in the middle, between the orange heads on the molecules in this picture, the yellow legs are lipid, like fat or oil. And this is why the skin is a barrier, because water from the outside of the cell can't pass through the middle of the membrane, the oil part, and get inside the cell. But what they couldn't see in the electron microscope is this. There were individual proteins built into the membrane. And there were two classes of proteins. One class of proteins is called receptors. It's a protein molecule that has an antenna on it, like a miniature television antenna. The other class of molecules are called channels. And these are molecules that are closed during their normal structure, like a fist, like closed. But when the signal comes and changes the structure, the channel opens up and makes a pore, so things can go in and out of the cell. And when the signal's gone, the protein goes back to the original shape, which is closed. So that's called a channel. So what was not visible in the electron microscope is there are proteins built into the membrane that are called receptors and channels. Well, the function of a receptor is to read an environmental signal. So, number one, whatever is coming from the environment, if it complements the vibration of the antenna on the receptor protein, it causes the receptor protein to change shape. And that's when the receptor can hook to the second protein called the channel. Well, when the receptor is not hooked to the channel is closed. But the moment an active receptor binds to the channel opens up. And therefore, when it's open, signals can go into the cell. And those signals control the behavior of the proteins inside the cell. And those signals can go into the nucleus and control genetic activity. So the function of the receptor is to respond to the signals. So that's where the stimulus comes in. The function of the channel is to make a response. So together, a receptor channel represents stimulus response. Another way of describing their function is a receptor represents an input, and the channel is an output, an I/O, which in computer and information technology represents a bit of data. Well, for me, this is important data bits built in the membrane because a receptor channel complex reads an environmental signal and sends that signal across the membrane to engage the functions of the cell, be it behavior or genetic activity. So this new science, as we talked about, is called signal transduction. And so what will happen is simply this. As you can see in the next illustration, a signal comes from the environment and binds to the surface of a membrane where there are receptors. Then the signal is relayed from one protein to the next protein. Like the story of dominoes, where one domino falls and it hits the other domino and that falls and they all fall. So there's a whole domino ring of dominoes falling down. Once the signal starts, it's a cascade. And those movements of the proteins that result from the signal create the behavior of the cell. And therefore, when we see and follow this path, what we're studying is called signal transduction. So this is how a signal from the environment generates behavior. Behavior doesn't act on its own. Behavior is driven by environmental signals. And so the membrane is the interface that takes the environmental signal and translates it into behavior of the proteins inside the cell. But then, as I mentioned, what happens is sometimes the protein's not there. And I say, well, then what happens? I say, well, then the signal goes from the membrane through the proteins in the cytoplasm, but then the signal enters the nucleus of the cell. And when that signal is in the nucleus of the cell, that signal controls gene activity. So signal controls gene activity. Environment controls gene activity, not genes controlling themselves. That's the old belief. The new understanding of how environmental signals control gene activity is the new science called Epigenetic Control. That new science was given a name in the 1990s. By then, I had already been working 25 years on the science of epigenetics, but we didn't have a name for it. But now there's a name, epigenetics, and it's a science of how environment controls genes. And what we were just tracing is that the signals from the environment go through the membrane, are connected from one protein to the next protein and follows through until they enter into the nucleus. And then in the nucleus, the signal activates gene activity. And this is a story of how environment controls genes. But then I said, but how does this actually work? And I say, well, then you go into the nucleus of the cell. And when you go into the nucleus of the cell, guess what? You find chromosomes. And there are 23 chromosomes from your mother and 23 chromosomes from your father, together 46 chromosomes. Interestingly illustrated in this slide where the pairs of chromosomes are staying different colors. So you can see a pair from the mother by finding like the two red ones or two matching colors. They represent one chromosome from the mother and one from the father. But the chromosomes are made out of 50% DNA and 50% protein. Well, we know the DNA is the blueprint to make the proteins, but what is the protein function in the chromosome? 50% of the chromosome is protein. Well, it turns out like this. The function of the protein is that it forms a sleeve around the DNA. And then in the center of this sleeve is a core of DNA genes lined up. And I say, well, how does that work? And here's how it works. It's simply like this, to give you a simple analogy. Let's say my arm is a chromosome, okay? And I say that my sleeve here, my sleeve is the whole chromosome. And I say the sleeve part is the protein, and underneath the protein is my bare arm. This would be the DNA. So let's say I write the genetic code for blue eyes on my arm using a marking pen. I say, can you read the code, the gene for blue eyes? And of course, it's got all the T's, C, A's, and G's, the subunits of the DNA, the code written on my arm. I say, can you read it? And you say, of course I can read that. I say, yeah, but what does it look like when it's back in the nucleus? It's not plain DNA. No, it's covered by a sleeve. I say, well, can you read the gene for DNA? I say, no, it's underneath the sleeve. I say, well, how can you read the gene? You have to take the sleeve off. I say, what's a sleeve made out of? Protein. I go, And what causes a protein to change shape? An environmental signal. So when the signal comes from the environment, it goes through the membrane, goes through the proteins, ends up in the nucleus, the signal binds to the protein on top of the DNA, the protein of the chromosome. And when the signal binds, it causes the protein sleeve to come off, and then guess what? Now I can read the gene. So the whole story of how genes work is illustrated in the next little video, and this little video is an animation, and what it really shows simply this is that a chromosome is a sleeve of protein inside of which is a core of DNA, which are the genes. At the beginning of each separate gene, there's a special protein built into the sleeve called a regulatory protein. And this regulatory protein is what is attached to the environmental signal when it comes in from outside. And when the signal binds to the regulatory protein, it causes the sleeve to pop off. So as you can see in this video, as the signal comes in, the signal binds to very specifically the regulatory protein of the protein that we need, that gene. And once the signal binds to the protein, the sleeve pops off. And now the DNA blueprint is exposed. And then I say, then a signal comes from the protein that copies that DNA, and that protein is called a polymerase. It makes a copy of the DNA. But this one is an RNA polymerase, meaning when it copies the DNA, it makes an RNA version, like a photocopy, like a Xerox copy of the gene. The RNA is the blueprint that goes into the cell, and the RNA is used to make the protein. And once the blueprint is made, I don't need the gene, so guess what? The signal pops off of the regulatory protein, and the sleeve goes back on, and the gene is covered up. Now, we can watch it again without the titles and see how quickly this occurs, and that is simply this. A signal comes in through the nucleus, binds to a protein on the sleeve of the chromosome. It causes that part of the sleeve to come off, exposes inside the DNA code for a protein. Then a protein enzyme called RNA polymerase comes up, copies the exposed protein, makes an RNA messenger molecule, which is the molecule used as a template, the blueprint, to make the protein. And once the RNA is made, the signal releases from the protein on the sleeve of the chromosome, and the sleeve comes back together again, and the gene is covered up. This is the mechanism of how genes work. Genes did not turn themselves on. Genes did not create the behavior. All of it is a result of the proteins. And these proteins respond to environmental signals. So the net result is a very important conclusion. Genes don't turn on and off by themselves. Genes are controlled by the signals from the environment. And that means if you control the environment, you then control which genes are going to be read. If you change the environment, you'll change which genes are going to be read. And this becomes very important because it simply says this. The old belief, genetic determinism, says genes control our life. The new belief, epigenetics, says no, environment and our perception of environment controls our life. And that's important because if genes control our life, then we're the victims of heredity. But if perception and environment control our lives, then we are the masters of our heredity because we're the ones that can change the environment and we can change our perception of the environment. Therefore, we control our genes and we control it with consciousness. The question is, how does epigenetics work? And the answer is very simple. Epigenetics are signals from the environment that come into the cell. Environmental signals then bind to the chromosome. The environmental signals can alter the DNA by adding what is called a methyl group, a small chemical group. And that generally marks that gene to be not used. It inhibits the gene when it gets methyl groups on it. That's usually what happens. So the environment can select which of the genes are going to be read by determining which ones get methyl groups. Whichever get methyl groups, those genes are not going to be read. And those without methyl groups can be read. So Epigenetics can determine which genes are going to be the active ones and which ones are not going to be active. But then number two, environmental signals can alter protein on the DNA. And we talk about, well, what are these proteins on the DNA? And the answer is simply this. We go back and look to see how a gene is made. And here's the story of the gene. When we see the DNA, the whole strand of DNA in a chromosome, 3% of that DNA is what represents genes. 97% of the DNA does not represent genes. 97% of the DNA has had the wrong name. It was called junk DNA because originally it was thought it was DNA that was just never used. Only 3% was being used and 97% was called junk. But now we know a different story. What was called junk DNA is some of the most important DNA in the whole world because that DNA is the one that regulates the function of the genes. Now, what I mean by that is following. Let's use an image of an old television set. And I say in the old days, a pattern would be sent by the broadcast station to the TV. It was called a test pattern. And I said, that's what you see on the screen, a test pattern. Well, a pattern is a gene. A gene is a pattern. So I say the broadcast is a gene. It makes a pattern. But then I say you can adjust some of the dials. You can turn it on and off. You can adjust the volume of how much. You can adjust the color. You can adjust the contrast, the focus. You can adjust the width or the height. So look, let me change some of the dials, and here's picture one. Look, I changed the picture. Most important point. Did I change the original broadcast? Is this the original broadcast the same, or did I change the broadcast by changing the dials on the television? The answer is no. Broadcast is still the same. It's just the dials on the television are changing the readout of the broadcast. Here, I'll change some more dials. Here's the picture is altered again. And yet, what? I changed the picture, but I didn't change the original program, the original pattern. I bring this up for a very important point. Epigenetics can change the readout of the gene without changing the genetic pattern. The genetic code can stay exactly the same, but I can change the readout of a gene. And so the significance is very important because it says environmental signals not only select a gene, but depending on the environmental signals, they can alter the protein made by the gene. Remember, the gene is the pattern. And along what is called the junk DNA, there are protein modules, they're called. This is the new finding. It's not empty DNA. They contain protein modules. Well, the modules are the equivalent of the dials on the television set. So what's the point? 3% of the DNA in the nucleus represent gene blueprints. 97% of the DNA represents parts of the DNA that control the activity and the readout of the blueprints. Not only can they turn on a program to be read or turn off the program to be read. Epigenetics does that. But epigenetics can change the exact reading of that program. So the point about epigenetics is very important. Epigenetics can alter the chemistry of the DNA and determine which genes are going to be read and which ones are not going to be read. But when epigenetic signals change the protein modules on this 97% of the DNA that doesn't code for the genes, they adjust the readout of the genes. And the important point about all this is that just through environmental signals being different, you can chop up the blueprint of the gene and create many different variations of proteins from the same blueprint by cutting the blueprint and taping it back together in a different sequence. And so evolution of the genetic activity is a result of environmental signals that can take a gene blueprint and copy it, making a messenger RNA, but then take that messenger RNA and using scissors and tape, they can cut the blueprint into pieces and then tape it together in a different sequence so I have a completely different readout. So the same blueprint can be cut up and pasted together in different ways so that I can actually create over 3,000 different protein molecules from the same gene blueprint based on the signals from the environment. So epigenetics has not changed the original code of the gene, but through environmental information, epigenetics can adjust the readout of the genes and create thousands of variations of proteins from each gene. Now, when you bring that story up, it's important to recognize some of the variations introduced by epigenetics might be beneficial, and some of the information introduced via epigenetics can be very destructive. Epigenetics just changes the protein. It's the environment that determines how that protein is going to be changed. So, for example, in my next illustration, there are two mice illustrated here. The one on the left, the blonde-colored mouse, that mouse is a lot larger and fatter than the normal-looking mouse on the right-hand side. The interesting point is the big orangey-looking mouse is called an agouti mutant. It has a mutation in it that causes changes in the body, such as the body is obese. The fur color is different. The mouse has diabetes and cardiovascular disease and dies very young. And that's the result of the agouti mutation. Now, the mouse on the right is a normal-looking mouse, but guess what? It's the same genetic identity as the mouse on the left, but it looked normal. I said, well, it's the same genes. How could it do that? And the answer is this. During a certain time in gestation, that means when the mouse was developing inside the womb, during a certain window of time during that development, the mouse was fed very specific diet containing things such as choline or vitamin B12, elements like that, at a very small time in the window of development. Let's say the mouse is developing over this entire time. I say only in a small window in development, just around here, for a few hours, if you give the mouse a diet containing such as the choline or the vitamin B12, it causes an epigenetic covering up of the mutant gene. So what's the point? The two mice have exactly the same genes, but on the mouse on the right, an environmental alteration by feeding those certain elements at a very certain time during development caused an epigenetic covering up of the gene, and the mouse is normal. But its genes are still the agouti mutant genes. If you mated this mouse, it would have a mutant offspring. So the point is very important. Epigenetics can change the readout of the gene, but not actually change the original gene. And in changing the readout, sometimes it's a benefit, and sometimes epigenetic changes actually are very toxic. So, for example, 90% of cancer has no connection with heredity. 90% of cancer is due to environment and lifestyle and belief issues. And I say, well, how did that create a cancer? And the answer was, the epigenetic alterations produced by those thoughts, those negative thoughts, produced a negative gene that caused a cancer. So all of a sudden it says the gene doesn't determine the character of your life. It is based on the environment and the experiences in your life that determine that. That's why buying a genetic readout may not tell you anything about the future at all, because all of your genes are quite changeable based on what's going on in your world at the time. And the next slide, it just shows a gene printout from two identical twins. Recognize this, at the moment identical twins are born, they have exactly the same genetics. But if you come back and read their gene readout year after year, you come back 5 years later and you read their gene readout, it's going to be different gene readout between them. If you come back 10 years later, the two twins are going to be further separated because each one's going to have completely different kinds of reading of their genes. One twin could end up dying of cancer and the other twin could live to 100 years old. And all of a sudden it says, well, then what was the difference? The answer was not the genes. They started with identical genes, but it was their life experiences and their beliefs and their environment that altered their genetic readout. And so the older they get and the more different their experiences, the more the twins will diverge and not have the same genetic readout. Epigenetics changes over time. It changes the genetic readout. The next story is a real interesting one from a researcher in San Francisco by the name of Dean Ornish. Now, he was working with prostate cancer patients. And what he did is he took his cancer patients and split them into two groups, A and B. And before they started the experiment, he read the genes of each group. So he had a genetic readout of these prostate cancer patients before the experiment. Now here's the experiment. Group A, all the participants got the exact same medical treatment that any prostate cancer patient would get, which includes drugs and radiation and all kinds of things like that. In contrast, Group B, all they did was change their diet, they learned how to meditate, and they also learned how to reduce stress in their life. And so both groups were in the experiment for 90 days. So they read the genes before 90 days, and then they read the genes of the participants in each group after 90 days. And here's what happened. In the group that got medical treatment, the same gene readout at the beginning was the gene readout at the end. The genes didn't change at all. But in those that changed lifestyle, changed diet, learned how to meditate and reduce stress, in 90 days, 500 genes changed their function. Most of these genes were connected with the prostate cancer. They were actually reversing the prostate cancer by changing lifestyle. And this fits the story. Epigenetics can cause the cancer, but epigenetics, responses to the environment and perceptions, can also cure the cancer. So epigenetics works in both ways. And how fast does this occur? In the next story, scientists measured 2 genes that concern inflammation in people who were undergoing meditation. So they read the genes, and then the people did 8 hours of meditation, and then they read the genes again. And guess what? The genes changed their function in 8 hours. The conclusion of this is very, very important, and it says this. The original story of genetics says that we're victims because genes determine our character, our emotions, and our behaviors, and genes turn on and off without us influencing them. Therefore, we're just programmed by genes. The new story of epigenetics reveals a different insight. It says that the environment, signals in the environment acting through the cell membrane, and then going into the nucleus via a cascade of one protein to the next protein into the nucleus. Once in the nucleus, these environmental signals adjust the reading and activity of the genes by adjusting the proteins that form the sleeve of the chromosome. And that you can adjust the readout of these genes and create thousands of variations based on whatever environmental signals come in, because these environmental signals adjust the protein modules, which are the equivalent of the dials on the television set. So epigenetics can turn a gene on and off, and epigenetics can say how much of a gene should be needed. Epigenetics can change the character of the gene by cutting it and pasting it back together in a different sequence. And the significance of all this is that it's controlled by the environment and our perception. Well, genetics, the old story, I'm a victim of the genes. Epigenetics, the new story, is I am a master of the genes. Because the control is not in the genes. The control is above the genes. The control is in consciousness and perception and nervous system. If you change your belief and your attitude and your environment, you can change the readout of the genes. In fact, just to conclude this section, it's important to define epigenetic control. The story started with genetic control, control by genes. But we end with epigenetic control. And I say, what does it mean? I say, epi means above. So, for example, the name of skin is called epidermis. It means the layer above the layer called the dermis, above dermis, epidermis. Now I say epigenetic control, and here's the fun part, because epigenetic control means control above the genes. The control is not in the genes, the control is in the consciousness of the individual. That's what controls our genes. And since we can control our consciousness, then we're not victims of our heredity. We are masters of our genetic expression. This is the third part of our discussion on heredity. In this part of the lecture, I'm going to talk about the nature of what are called telomeres. At the end of our DNA molecules, which are long threads, there's an extra length of DNA that doesn't code for anything. It's not a gene. It's not part of epigenetics. It's just an extra piece of DNA. And the question is, why do we have this extra piece of DNA on there, the so-called telomere? And the answer is very important, and it relates to this. DNA is stable as a double helix. But if the helix begins to unwind, then the free ends become attacked by enzymes and start to destroy the nature of the DNA. And the significance of this is that if the DNA was exposed at the ends, then enzymes would chop it all up and essentially destroy the gene blueprints as well. So to keep the ends from being attacked and from opening up, the extra ends on the DNA, the extra pieces called telomeres, act to hold the DNA double helix together. To give you a picture of what that is related to, consider a shoelace. It's made out of a lot of fibers of thread held together at the end by a plastic cap called the aglet in English. The aglet is keeping all the fibers from falling apart. As you can see in the right-hand picture, if the aglet comes off, the filaments of the shoelace start to fray, they start to unfold, and now the shoelace is very difficult to put into the little tiny hole in the shoe. Its function is interfered with. Well, the same thing occurs with DNA. At the end of DNA, these extra lengths of DNA called telomeres are the equivalent of an aglet. They hold the DNA from coming apart, and as a result keep it from being broken down by enzymes. So telomeres are safety pieces on the end of DNA to prevent them from being enzymatically broken down. Now, to see where they are, here's a picture first of a nucleus opened up with the chromosomes exposed. And again, the chromosomes are DNA-covered sleeves with protein sleeves on the outside of the DNA. That if you want to see where the telomeres are, in the next slide, which is a higher magnification of an X chromosome and a Y chromosome, you can see where the telomeres are because they're stained red. They're at the very ends of the DNA strands and each end of the chromosome, as you would expect to find them. These telomeres, as I said, are protective devices that keep the DNA from unfolding and keep it stable. But there's another very important role for telomeres, and it has to do with this. When you want to copy the DNA, as in this illustration, an enzyme called the helicase enzyme splits the DNA double helix into single strands. And as the single strands are exposed, a protein called the DNA polymerase will land on the DNA. And like a train going down the track, the polymerase moves down the length of the DNA. And as it's moving down the length of the DNA, the polymerase is copying the genetic code to making a duplicate copy so that the double helix is returned. First we have a single helix, but after the polymerase moves down, it makes a second copy, and that completes the double helix. Well, as you can see, the enzyme moves down to the end of the DNA strand. And at the end of the strand, which is indicated in green, little bumps on there, those green bumps represent where the telomere is. The red part of the DNA strand is where the gene programs are. And I say, so what's relevant about this? I say the polymerase went down the length of the DNA and copied the duplicate right above it. And I say, and then what happens? I say, well, it gets to the end of the DNA, and I say, then what's the problem? As you see, when the polymerase pops off, it did not copy the little piece of DNA on which it was sitting. So every time you copy the DNA, you lose a short piece at the end where the DNA cannot be copied because the polymerase is sitting on top of it. And the next move, the polymerase falls off. So it says, every time you copy DNA, the new copy is a little bit shorter than the original copy. Well, this is very important because if I keep shortening the DNA and I shorten to the point that I'm cutting off the genetic code, the part in red, well, that means I'm going to ruin the blueprints by breaking it off and not having a complete blueprint when the DNA is copied. So the function of telomeres is to allow the DNA to be copied over and over again. And this, every time the DNA is copied, it gets shorter and shorter. But because the telomeres are there, I'm not cutting off part of the genetic code. So as long as there's telomeres, then I am actually allowing the copy of the DNA blueprint without destroying the blueprint. But now there's a problem, and the problem is this. If I'm losing a piece of telomere every time I copy the DNA, how many times can I copy that DNA before I'm actually now cutting into the gene program and losing part of the program. And the reason why this is important is telomere length is a predictor of aging, disease, and depression. As the telomeres get really, really short, that's when we start aging more. That's when diseases become more rampant because now we're interfering with the gene blueprint. And it's also associated with depression. So all of a sudden there was an idea that came to a scientist by the name of Leonard Hayflick. He said, if the telomere is only so long, how many times can I copy it before I lose the entire telomere and then we cut into the genes? I say, why is that time important? Because accordingly, that would be how long we could live. How many times can my cell divide before the DNA cannot be copied correctly? That will determine a lifespan. Well, Leonard Hayflick, the scientist, did the calculation and says that we should come out to live to near 100, 110 years of age, which is interesting because that's how old people seem to live. And it fits the story. Yeah, by around 110, you've copied the DNA on your stem cells. Remember, the stem cells are replacing your cells every day, and as a result, they have to keep dividing. And if you run out of stem cells, you can't replace your cells anymore. And as a result, at that point, aging and disease become apparent in the human body. So you want to keep your stem cells dividing, and yet Leonard Hayflick says they can only divide for a person until they're about 100, 110 years of age. I go, oh, so our life is fixed. And I go, no, because later a woman, Elizabeth Blackburn, discovered an enzyme called telomerase. And that enzyme is an enzyme that can extend the length of your telomeres. I go, oh, well, wait a minute then. Then there's not a fixed lifespan. Because if we can keep extending the telomeres, we can keep dividing the stem cells. And as long as we keep dividing the stem cells, we can stay alive. So scientists automatically have started to recognize that a minimum age for a human should be 150. And you say, well, why are we dying so young? And that's when the cool part comes in, because this is the part where the enzyme is working or not working. Here's the point. The enzyme can be engaged, which would then increase our life, keep us healthy for a lot longer. Or you can inhibit the enzyme and stop it from working, which means we age faster and get sick faster. So all of a sudden it says our life is really based on how much activity we can get out of telomerase. So I'm going to now list first those things that inhibit the telomerase enzyme. And the reason why this is important is because these things can shorten your life. And then I'll make a list of those things that will actually expand your telomerase activity and give you a longer life. So first list, these are the things that can shorten your life experience by causing the telomerase enzyme to not work. Number one, poor nutrition. Yes, if I'm building a machine and I'm not building it with a quality nutrition, there's a point where the machine will break down, and that breakdown is associated also with telomerase not working anymore. The other parts of this that are very significant are how we feel about being alive. So, for example, when a child gets abused, which could be either physical abuse, being hit, or now we know that verbal abuse is equally damaging. Yelling at a child is the same as beating up the child in regard to abuse. Abuse takes the joy out of life, and the child doesn't wish to be alive when it's abused. Guess what? The consciousness of being abused shuts down the telomerase activity. The activity is really connected to our desire to be alive or our desire not to be alive, and that's based on our life experiences. So another example is domestic violence. A person who lives under domestic violence does not wake up every day thinking, I want to be alive. They don't want to be alive, and guess what? Domestic violence can shut down the enzyme. Post-traumatic stress disorder, another version of stresses that continually impact our lives in a negative way, also shuts down the telomerase enzyme. One of the biggest things that influences our telomerase activity is whether we have self-love, meaning we love ourselves, and if we are loved by others as well. So when there's no self-love, And that happens to almost all of us for a very particular reason, because during our child development, when we were kids, our parents used to criticize us all the time to try to get us to do better. And they would say critical things like, oh, you're not smart enough, you don't deserve something, that's not good enough, you can do better. These are criticisms. And a child records those criticisms, and if we become so critical of ourselves, which most people are, 80 to 90% of us are so critical, that we don't love ourselves. Because the first thing is, when we think of ourselves, are all the critical problems that we have. We're not lovable, we're not deserving, we're not good enough. And so most people, 80% or more people, don't have any self-love because they have such critical review of their own life. And if you're in a loving relationship and that falls apart, loss of love as well as no self-love, both will inhibit the enzyme and shorten our life experience on this planet. And perhaps the biggest thing that affects the enzyme is purpose. When people retire and they have no purpose, or people have no job or no vision, no future, and they have nothing to look forward to, the enzyme stops working. Telomerase stops working. Why? Well, if you have nothing to look forward to, then why do you want to stay alive longer? Think about it that way. That thought is what shuts down the enzyme. So the telomerase enzyme can be inhibited by things that make our lives very unhappy or angry or sad or scared, violence in our life, lack of love in our life, lack of anything to do, such as when retired people retire and they have no place to go. Guess what? They die very quickly after retirement. Why? Because they had nothing to do. And therefore, the feedback from the mind to the genes via epigenetics controls telomerase activity. If I say I have nothing to do, then why would I activate the enzyme? Henceforth, I interfere with the enzyme. So those are the things that interfere and shorten our lives. But now let's go to the list of things that increase telomerase activity, because that's the one that we want. That means we can stay healthier, longer, and have a more wonderful life. So, number one, first, like we started off with before, nutrition is very important. So good nutrition is feeding the body and keeping it in high performance and high running order. And in that case, the telomerase is going to keep us going because the machine's running really good. Good nutrition includes especially omega-3s in our diet, which are very important in engaging telomerase activity. Another thing that extends our life is exercise. Yes, we're using our body in exercise. And what are we feeding back information? I want my body to be strong. I want it to work. So I'm exercising and I'm keeping it alive and I'm keeping it in top form. Well, if that's our intention to keep it alive and keep it in top form, that intention is also which activates epigenetically telomerase activity. Then there are things that we love about life so much that we want more life. So, for example, things such as happiness or gratitude or having a positive outlook enhance telomerase activity, because those things are feedback saying, I love my life, I want more life, and I'm looking forward to it. Your mind is actually then going to epigenetically activate telomerase and keep you healthier and happier with a longer life. Love, self-love, again, these are the most important things. If we love ourselves, then we want to be here. If we don't love ourselves, it's like, it's a waste of time. So love of yourself becomes very important in activating telomerase and keeping you alive longer, because you love your life experience. You want more of your life experiences. And lastly, being in service is profoundly important. Is there a reason why you wake up every day? Is there something you have to do? Caregivers live much healthier, longer lives than people who are not caregivers. Why? Because consciously they know, I have to stay healthy. Why? I have to take care of somebody else. So therefore, I have to be healthier longer to take care of somebody. So a caregiver is giving epigenetic information to activate telomerase because they have something to do. Again, this is why retired people, their lives are shorter, because once they run out of something to do, they have no feedback, no reason to activate telomerase activity. So in conclusion, and this part is very important, it says that the DNA double helix is stabilized by extra pieces of DNA called telomeres. Telomeres are lost piece by piece every time the cell reproduces. And if you lose the telomeres completely, every time you copy the DNA after that, every time you copy it, you start copying and cutting off part of the gene program, and that's what causes aging and disease. But there's an enzyme called telomerase, which can extend the telomeres, make them longer. What does that mean? Well, directly it means that we'd have a longer, healthier life. And there are factors that activate that enzyme and factors that inhibit that enzyme, and basically they're split into two categories. Those factors that say, I love my life and I'm enjoying my life and I have things to do in my life and I'm taking care of myself, those activities engage telomerase and give us a longer life and a healthier life. In contrast, when we don't love ourselves, we're critical of our own existence on this planet, when we have nothing to do, no purpose in life, and we're abused and we find trauma and trouble and we don't want any more of this, what are we feeding back to our system? Our consciousness is saying epigenetically, I think I've had enough life, no more. And as a result of epigenetics of a negative type, we can shut down the telomerase activity and then actually shorten our life and increase the amount of disease and depression that we experience. So there's an important conclusion in this, and that is heredity does not say there's a precise length of life. Life can be variable based on how much we are enjoying our life and how much we can eliminate all the negative perceptions and beliefs of the environment in which we live. And so that's a mission. We can increase our life if we want to do it, and there are ways to do it, and all of them involve being happy, loving yourself, and taking care of the body that you received. Thank you.